Hi, everyone. This is Dr. Nicole Quinn, and my co-host today is my friend and international travel buddy, Dr. Jason Goldsmith, co-host of the Immunology podcast. Welcome to the Lab Coats in Life podcast, where we help scientists thrive. The Lab Coats in Life podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life sciences research and fostering communication and collaboration in science. If you enjoy the Lab Coats in Life podcast, rate us and leave a review. You can also suggest ideas or recommend guests for new episodes. Today, we have Dr. Alex Shalek from MIT on the podcast to talk about open data in science. Specifically, Alex is here to share insights from his experience creating technologies and methods achievable by any lab anywhere in the world through open source experimental protocols and computational packages. Dr. Shalek completed his BA in chemical physics from Columbia and his PhD in chemical physics from Harvard. I had the pleasure of sitting as a co-panelist with Alex and others recently at the IUIS meeting in Cape Town, South Africa, where we discussed open science. Admittedly, I went into that conversation thinking more about science communication, namely open access publishing and preprints, whereas Alex was providing expertise on sharing data specifically. I was fascinated and intrigued and humbled by his insights and his eloquent way of framing some of the opportunities and challenges surrounding open data. So I'm very excited to have him here on Lab Coats in Life with Jason, who is also at IUIS, to dig further into this topic. But before we get to that, did you know that the Lab Coats in Life podcast is part of STEMCELL Technologies mentorship resources? Would you like to learn more about practical soft skills to help you communicate your science and read up on current trends and efforts in the scientific community? Visit www.stemcell.com/labcoatsinlife to find articles, free downloadable checklists, and more to help you thrive in science. Alex, welcome to the podcast. Jason, great to see you again.
Good to see you.
Thank you both for having me.
Welcome. Okay, I'm going to start by defining what we're talking about with respect to open data. So, like I said in the intro, we're specifically not talking about open access publishing or sharing final polished stories. We're talking about sharing raw data, raw methods, and doing that on openly accessible platforms. So I will admit that my experience in this is pretty naive. I shared a few DNA sequences from salmon hemoglobin way back when I was doing my research, my grad research like 10 years ago. But things have obviously changed a whole lot since then. And I think open sharing has moved well beyond genomics. So maybe Alex and then Jason, can you speak to your specific experiences with this now and how you're currently working within this open data sharing world?
So it's a really big question. I think that one of the things that's so exciting about genomics is the promise that it enables you to explore many different hypotheses. And more than thinking about the things that you can do in your own experiment is the idea that as you bring together data sets across multiple different experiments, there'll be opportunities for reanalysis that will enable emergent insights. And so I think it's one of those things where we always think that as we get more and more data, there's going to be real opportunities to take advantage of that, to push the boundaries of what we know, to learn new things about how cells work, about how our tissues work, about what drives disease, and incredible opportunities for technology development, whether it's understanding what we need experimentally or building new computational approaches. I'd say that the recognition of the importance of data also underscores some of the complexities associated with it, because many people are interested in having access to data. And many people have differences in levels of access to the data. And in many places, when we originally envisioned the studies and got consents for them, particularly if we're thinking about human data, some of the use cases weren't always envisioned in the way in which we might want to use them now. And it opens up lots of questions about how to best do open access data and how to do the kinds of things that we all can see as being potentially incredibly powerful, while at the same time recognizing that fundamentally, we owe respect to the individuals who partnered with us in these studies by sharing samples and sharing material at a time when things may have been very precarious or very difficult for them personally. And so there's this tension between the promise and between the responsibilities that we have as investigators, but the responsibility that we also have transitively as people that are getting involved in reanalyzing those samples and making sure that we do as well as we can by those individuals and their wishes.
Yeah, there's so much in there, and I think we're going to dig into all of those. Well, maybe not all of them, but some of those big picture ideas and some of the caveats and concerns. Jason, where are you sitting? You know, you're coming at this from an industry perspective, whereas Alex is working in as an academic, and I think there's different thoughts and considerations around open data.
So, I'll say the mercenary answer is that industry loves it if we don't have to provide it, right? I mean, so fundamentally, we see this in papers all the time, academic papers, right? Hey, I have this new interesting finding, and I went to a data set of human samples and saw it there, and so that's I think the gold standard example Alex is getting at of like new discoveries from old data. You don't have to go run a new trial if the consent's all clean. There's a lot there, but by the same token, when you start getting private industry concerns involved, like private industry spends millions of dollars of investor income or public stockholder income, right? But people's money to generate these data sets. And so they have an interest in seeing them get the benefits of that. And if you go and share all of the data out immediately, and they usually share the data at some point after the trial's out and there's publishing, but it could have a decade plus embargo on it because they want to mine all that information and be the beneficiaries of it. And they actually have a responsibility because of how the investment structure works to do that. And so it creates this interesting dichotomy where I think industry like broadly loves public data that's available. Taxpayers pay data and that data is available for everyone to use. Companies are taxpayers too, but then they have an interest in not sharing their private data sets often. I mean, I think COVID's an example to the contrary, but even then, the federal government kind of backstopped the financial gain mechanism. They said, hey, if you can get a vaccine out or 2 vaccines or 3, we'll buy them all. You just get them approved. And so everyone's like, yeah, we'll share data with each other to get these all approved. But they removed the disincentive not to share. And so I think that's part of the complexity from an industry perspective is a lot of stuff, if you put in a patent or you put it in a paper, or you put it in a trademark or not a trademark, but a copyright, any of that, it's now freely available. Whereas if you keep it secret, it's a trade secret and not available. And that has strategic advantage to companies.
So I totally hear you, Jason. I would say that we're looking at it very similarly. It's just that we have different groups of people to whom we're responsible. And some of the things that you do in terms of setting up your cohorts, thinking about consents, working through it, like there are different things that you're committing to those communities as part of that process. And particularly as I think about it as an academic, very often we don't have the same ability to translate insights into products that can impact people in the way in which you want. Like, as much as I'd love to be the person that develops a drug, brings it to market, and delivers it to an individual to really alleviate suffering, in many places, that's not what the academic model is made to do. It's made to find those targets, to find maybe a lead compound, and then to move it out to industry. And so I'd say that we have different responsibilities to people, and therefore, we can think about data in different ways. But I would say that there's also an importance in having open data. And this came up, in the meeting that we had at IUIS that Joachim Schulze was talking about, which is that in many places, when we think about doing a study or, repeating a study or setting it up again, A, it's not a very efficient use of money. B, it's not a very efficient use of time. And C, there are tons of hidden variables in the way in each of us thinks about structuring our studies and building stuff out, where there can be biases or confounders of which we're not totally aware. And it shows up in a lot of places where people think about like the gut microbiome in different mouse colonies leading to different results. But I think that sometimes it's nice to have completely different studies structured by different people with different data sets where you can see glimpses of the hypotheses or the ideas that you had to give you that sanity check that tells you you're on the right path and you can move forward. So I think that there's an independent value to open data, which is that it gives us the opportunity to sniff test or to, sort of really fact check some of the things that come out of what we're doing in a way where, sometimes the data doesn't lie.
I definitely think so. I think the difficulty is in aligning those incentives properly, right? So this absolutely happens where in industry groups, 2 big companies are working on the same pathway and some of their people decide they should share data with each other and enter into agreement or a CDA and then share that because they see benefit in all those things you just said. But a little company like the one I work for, or take any example, may not want to share that because if they share it with the big company is going to go outspend them and essentially jump 8 steps. And what the little company wants to do is sell them that data, so to speak, right? And so that's where it gets very tough because all the benefits are there, but if the company's like spent $10 million of investor money to create the data, they want to see that 10 million back. Otherwise it doesn't exist anymore.
I want to see more than 10 million back.
I
And
know.
as the son of an IP attorney, I will sit here and say I totally understand. And the biggest question a lot of this is, what do you cover versus what is trade secret? What do you want to keep internal? And where do you really get the most bang for your buck out of all the things that you put in? Because that's critical. And we get to speak from different positions and approach this in different ways. I think we both see the utility in open data. I think that we just have to approach our engagement with it in different ways. And so, to your point, I might think about being a more proactive generator of open data and open sharing of data. But the thing that you're highlighting is your responsibility to many of the people that fund your company or that fund companies. And I would say that when I generate open data, I also have to think about who I partner with, because those individuals might have different desires and wishes. When I partner with individuals that are in the global south and that have less access to some of the computational infrastructure or don't have the same resources and know how to move quickly, there are large conversations about should that data become open immediately, or do we provide protected periods over which they can engage with the data? How do we make sure that the researchers that are working on that actually generate the greatest return on that? And how do we make sure that that return accrues back to the industry? Back to the community. As you think through these sorts of things, like open data is great, but at the same time, we have to think about who owns that data. And that's the people that are involved in providing the samples that generate the data, but also the individuals who are involved in generating that data. And what are the things that are responsible to do with it? And we could take this sort of approach that says, well, if we took the samples and we brought them to Boston, where I'm sitting right now, Cambridge, and ran them and generated data, like it would accrue data benefit to the communities that donated the samples or that collaborated with us as partners by contributing samples. But that might be a little disingenuous if we're not really engaging those communities and thinking about the researchers in those communities and thinking about what local desires and wants are. And so, I would say I completely understand what you're saying. We just have a different group of stakeholders, and the incentives to open science aren't the same among all of those groups. And so, as much as I appreciate the strong push to share, and particularly from government organizations that I'm affiliated with, and as much as I believe in sharing, I recognize that I sit in a position of privilege and that I'm able to do that. Being careful at sometimes to not put some of my students or my trainees at risk because they have careers and they have things that they need. But I can do so much more easily and much more freely than many people around the world. And it's not fair for me to assume that every single person can be as open with their data and share it as freely, even when we're all working towards something that we agree is a major goal, like figuring out how to fight COVID-19 or tuberculosis or HIV. And so I'd say that what's really important in understanding open data is understanding who's involved in the data in terms of the donation of the samples, the generation of the data, the analysis of the data, and what are the responsibilities that go with that entire process. And so I'd even say it's different. It's not just academia or industry. It involves really thinking comprehensively about who's implicated and what their desires and wishes are, and what that means is a responsible way to engage.
Alex, this is exactly why I invited you on this podcast, and why your name came to mind immediately when we came up with this, is because I went into that panel really, really naive with this lofty, like, well, we're all here for the benefit of science, and we have to make the sacrifices and align on this, even if there's individual sacrifices or profitability sacrifices or whatever it is, we have to set up the apparatus or the infrastructure of science in a way that we can move towards this open data model, because it will accelerate science, because it will remove barriers, because we can maybe address the reproducibility crisis. We can get into all the benefits of open data. And you kind of looked over at me and you were like, yeah, Nicole, okay, great. You have you have all of these big ideas, but there are so many nuances there. And that's what you've kind of dug into right away. And so I want to back up a little bit and go back into, what is the perfect that's a really big question, but where do we want to go with this? Like it's changed so much in the last decade that, let's catch people up. What would be the ideal state for data, and what would be sort of the checks and balances that we could put in place so that we can combat some things around privacy, around reproducibility, and then we can kind of come back and dig into, are there examples of when this has worked, or is this completely naive of us? I don't know. Alex, can you speak to any of that?
I can, and you can cut me off when you want to transition to something else. I would say that we have to separate out the reproducibility piece from the responsibility piece, because I see them as very different things. I can think about how to create reproducibility and what's required to create reproducibility and how open data can contribute to that. And I think about that as being different than some of the other pieces that we're talking about. I think in many places, issues with reproducibility come down to the fact that we very often don't methodically and systematically characterize a lot of the things that go into experiments. We don't necessarily pay attention to them. They're the hidden variables. And so they're things that sort of, that we just take for granted, but we don't realize that other investigators don't take the same things for granted, or some of the things that we think of as being, unimportant turn out to be important when you look in different contexts, right? And so I think that a lot of figuring out the reproducibility piece is finding a better language, a better way of communicating. One of the big things that people talk a lot about when they talk about open data is this idea of creating better standards, better metadata, better references, better ways of annotating things to enable greater consistency and sharing of information. I think that one of the things that I really dislike about publishing is how you have to really pack all of your methods and approaches into the smallest place possible with the fewest words possible, because you're trying to get into a print format. And I, love that there are some journals that try and focus on methodological approaches where you can talk about some of these pieces more freely. But I think we need to do a better job of describing what we've done, how we've done it, why we've done it, what the assumptions were behind it, so that people can have those conversations. And if you're not creating a format where people can easily look at that and add it, particularly as they're amalgamating the information from lots of different studies, there's a tremendous opportunity to find that things aren't reproducible for reasons that are totally explainable. And so you could say, oh, this isn't reproducible because these people did this in serum-free media, whereas these people did this in media with serum. And you would say, no, it's entirely biologically predictable. The results actually reproduce biological features. They just don't reproduce the same results because they were done in different ways, in ways that make sense. And so on the reproducibility side, I'd say that a lot of where I think that efforts towards open data can be important is in creating standardization of descriptions of experimental approaches, of metadata, of ways of people going back and forth, of normalizing the idea of having conversations. I mean, obviously, there's this point around trade secret that Jason brought up before that comes up in some context, even within academic labs, but you really need greater exchange.
Well, I think actually reproducibility is the area that industry cares the most about in a couple ways. I think one example that we all know about is clinical testing, right? So if I go get a blood count and it's at one hospital or another hospital or even in another country, often those are medical devices that go through really rigorous reproducibility. And obviously, every experiment by a scientist can't be that way. But regulators set pretty strict guidelines about how you have to describe your assay and the level of detail of it, whether that's for one of those clinical devices that become a medical device, or to measure how much Tylenol is in your Tylenol pill, right? That is a rigorously reproducible measurement. And industry will take that same technology for ibuprofen and for Lipitor and anything else, right? And so that may be an area where lessons learned from regulators on how to describe assays precisely and what standards are needed for robust reproducibility, or at least the descriptions therein. You're not going to run 100 samples in academia. but you can describe it, as to your point, better than we do. And I think there's a really good point there that like, is it serum free media or is it with serum? And they don't write that down, and like, oh, you need to angle the thing at 45 degrees. But in pharma, that's all really regulated to like the temperature of the pipette and the angle of it and what brand. And that's how they get around that thing, because the FDA wants it to work every single time you measure the Tylenol in your pill. And there could be some lessons from there actually that could help academia and publication in general in terms of conveying that information, even if you don't have to do it to the same level, because that wouldn't be appropriate for the same level, but to pass that detail on.
So I agree and I disagree, Jason, because like we're talking about things that could be similar or could be different. The idea of a mass, it's consistent. We have a scale that we use to measure mass. We can be very reproducible in how we approach it. Temperature, something where we have a scale. I was teaching it, in a class earlier today where temperature scales. And so something where we can be very consistent. I think the problem is that when you think about biology and the kinds of big data we're talking about in genomics, right, you have lots of things that you cannot control, right? We engage with different groups of individuals as donors and partners based upon what study we're doing. Those people aren't identical. They don't have identical lived experiences. They don't have things that would enable you to reestablish the same system. And even if you think about it with mice, or pick your favorite model system, it is hard to create something that is exactly identical so that you can go over and over again and have the same instance so that you can get the same output over and over again in exactly the same way. And so what becomes important is to understand what drives variability and to really use that to think about whether or not what you're seeing is consistent. And I think that what you really want to say when you say reproducible is the biological mechanism consistent, as opposed to did we get exactly the same results? The entire way I got into single cell genomics was because we took cells that were supposed to be identical. We hit them with exactly the same stimulus and they all responded differently. And we tried to take a system that was supposed to be as identical as was humanly possible. Post-mitotic cells synchronized in a way, hit with a bacterial Armageddon, and they responded differently. Now, those differences in response gave us something to correlate against, which helped us figure out mechanisms that we could then go back and validate. But I think that the point is that biology has some variability in it. And as a physicist, I'm not going to sit here and tell you it's quantumness of biology, but there is some variability, and we want to understand whether or not that variability is a biological feature or a technical feature. And so I would separate out the reproducibility piece into technical reproducibility and biological reproducibility, and 100% agree with you on the technical piece.
Yeah, I mean, it's just writing it down. Like, to your point, how often in the methods do you see when you collect samples from donors, how many hours until it was frozen? They'll have some range. Samples were frozen within 85 hours. And like, well, that's like, what does that mean? Is it most of them within 24, 70? Like, how do you?
It's so important. I know from some of the stuff where we were involved in rapid autopsy work during the COVID-19 pandemic, it was one of those things where the amount of time from sample collection to processing, or from when the individual unfortunately passed away from COVID to when the sample was collected, the temperatures that it was kept at, and like all these things play a critical role in driving things, and they're understandable. You can see why they do specific things and why you get specific results, but only in light of having that information. And in many places, we aren't careful enough in writing it down. And I think we have to create standards around that being an expectation, because it leads to much better interpretability. And I want to maybe think about reproducibility versus interpretability. Consistent interpretation, I think, is what we want to go for, particularly as we move to biology, as opposed to technical reproducibility, which is a must in order to get to a place where we have biological consistency.
And that's where I think regulators have done a little bit, is they've kind of created like, hey, if you're going to submit this, you have to tell us the time you stored it at and the average and the mean. And like they're, because of the stakes on, say, drug potency and manufacturing of sterile things, all that, they have kind of created a playbook of important variables. I think it's not going to translate 100%, but you could learn from to an extent with what regulators have demanded of pharmaceutical assays, just in terms of a playbook of write it down, please.
So my question there is, Jason, I think you make a great point that the FDA is, or governing bodies have created these standards and communicated these standards. Who does that for academia? Where does that come from, and how does it get implemented? And what are these databases? Like, who's running them? Who's overseeing them?
So this is where it gets so interesting, because Jason, like, there is, and Nicole, there is an incentive in industry for this repeatability. You want to do no harm by the person who's going to take that Tylenol or the aspirin, and there are lots of repercussions to that not happening. So not only is it the right thing to do, you wouldn't want to have something that was adverse for all the reasons in the universe. When you think about it in academia, there's a lot of incentive, to do it within your own lab and to be very consistent, to produce results that are very consistent, and to think about how to do things in a way that provide validation, right? And we like to use multiple different methods, multiple different approaches, multiple different systems, like as many pieces of information as we possibly can get. But now, as you think about open data, where you're pulling together resources across labs, that's where I think, Nicole, it gets really hard, because the question is, who oversees that? Who convinces you that you should be adhering to specific standards, gets you to intellectually buy into the value of doing that? And particularly as it becomes a period where funding decreases or stays the same and inflation goes up, it becomes really hard to think about about doing extra work and putting extra onus on people. And so I think that's the rub. I think a lot of it comes down to people that are big proponents and advocates of the possibilities of big data, or people that want to do work that requires big data to sort of push these things forward. And so I think it's a lot of convincing and getting people to the table and telling them they have to do this. But I think that it's hard to say this is what will incentivize people to adopt a common standard versus adopt best practices, because I think everybody wants best practices. They want to do good science. They want to make sure that what they're putting out into the universe is high quality, and it's not going to come back to them that something was wrong or they need to retract the paper, because nobody ever wants that.
Yeah. That I want to come back to something you said earlier, when I was talking about my naivety and how I was like, it's all for the greater good and we should do it. And one of the points you made was, yes, and I'm also obligated to, as a PI and as a mentor in a lab, to advocate for my students and protect their knowledge and allow them, um, the best possible career out of this. And it's one thing to sit in a place of a tenured professor or somebody who's employed in biotech to say we should all be doing this for the greater good, but it's a different thing to say to trainees. And I don't know if I'm specifically representing what you said accurately, but it stuck with me. So I'm wondering if you could.
It's critical, and I think that this idea of sharing and when to share is really important, because, as you say, I sit in a position of privilege. I'm a tenured professor. Every single paper is important to me, but it is not the piece that makes my career, that leads to my PhD, that lines me up for a postdoc, or that might line me up for a job, right? So I get to think about it slightly differently. I think that in general, I want to get back to something that comes up a little bit earlier. You're talking about the greater good, and I think that you ask, why do we do these things and what motivates us? And I know that for me personally, I want to help people. And what I have to do when I think about that, like, I do this job because I want to help people. And that means I want to find causes for illnesses. I want to push towards creating better therapies. I want to, mentor and train people. That's why I'm in academia, right? And so I have to think about who I'm trying to, do good for, and who I'm responsible to. And I think that as we think about open data, early sharing can potentially put some of my trainees at a disadvantage, because they don't have the time to do their analysis, write up their paper, work through things. I think that what becomes critical in sharing early and sharing openly is to set expectations, to have conversations, to put people in a place where they're comfortable with that, and where you basically have created agreements that protect individuals, right? I remember, it's really funny, during COVID-19, we had all these partnerships where we work with people all around the globe. And we wanted to show for multiple different species, multiple different tissues, multiple different contexts, what were the cells that were most likely to express ACE2 and TMPRSS2. So likely targets of COVID, SARS-CoV-2 in COVID-19. And I had to have a series of conversations with the editors, saying like, I can't share all of this data because these people haven't published, but what I can do is I can tell you all the cell types and I can give you the expression of these two genes or this set of genes, all the potential entry factors plus ACE2 and tell you what the cell types are and maybe give you a marker or two. But there were things where we had to think about what we were going to give and what we're going to hold back. And in other places, we can think about sharing data early, but we can have agreements. We can set things up in the same way that companies would set up NDAs where we agree that we'll share this early, but you'll wait for us to, put our paper up on bioRxiv and then cite it. But I think that what I've learned is that if we're very careful and thoughtful and we have the hard conversations early, right? It puts us in a better position where we can take care of the people to whom we're beholden. And it's not a bad thing for me to say, like, look, I trust you, but I need to say this to you because I represent this trainee, and this trainee has put a lot of trust and a lot of faith in me. And it is my job to respect that trainee and to have this conversation so we're on the same page ahead of time. And so I've just learned to be very proactive in those conversations and to recognize that every time I set up a collaboration or a partnership, I need to understand what the other PI wants, what the other student wants, what my student wants. And there are places where some things work for me, but they don't necessarily work for everybody involved. And you have to just have the maturity to say, this is a place where we have to step away. And so this gets back to the larger conversation about, about open data more broadly. There's a lot of people that push for open data, but I think that when we think about communities and we think about those that are involved in generating and using data, we have to take the time to think about data benefit, right? And data benefit to me, this is one of these things we talk about all the time. We don't really think about who the data benefits and how. And so I like to think a lot about the communities that are impacted by whatever we're studying, right? And that's not just the people within science. So it could be the trainees, the PIs that need those papers to get grants or to graduate, but also the individuals that might be impacted by a specific disease and what they hope and what they want. And at the same time, you have to balance, who in our lab can do it, what are the things that they need, what stage of their career at. And so I think being careful and having very direct conversations about it becomes important. And I'm not going to say that, I was always as thoughtful about this, but I've learned along the way to be very proactive and to be very careful to make sure that I understand what people want and to constantly check in to make sure that I really do understand it hasn't evolved in a way that's going to become problematic.
Mm-hmm. I want to let Jason talk, but I just want to give a comment that that last comment that you made, that you said you haven't always been this way. The whole time you were talking, I was like, well, this is an experienced MIT professor who's been here for, doing this stuff for a while. How do we provide, this isn't a question, it's kind of rhetorical, but that kind of information and knowledge and those skills of having, frameworks for having those conversations, those need to get out there. People need to be thinking about things. People who've never, people like me who are totally naive to this we need to be having these conversations so people understand those caveats and nuances.
So 100% agree. Podcasts like this are a great way of starting those conversations. And it's funny, when I talk to my trainees, I always say things like, the things that got me to this position are not the things that position me for success in this position, right? I was selected in because of what I was able to accomplish with others in science and because some of the ideas I had. But really now I run a little company, right? And I have to think about, my trainees, not employees, because really I work for them, but I have to think about the people with whom I work and, what they need. And so there are things that I wish I had done earlier, like leadership training. And I push my students, and whenever there's opportunities, I try and get them involved in it. And there's other pieces of the puzzle. And I think that making that part of graduate curriculum, like creating all those sorts of soft skill things that become critical, regardless of whether you go into academic science or to, industrial science or completely leave science, to do something different. I think we have to make that more part of the practice, because I went in, naive in some places and with best intentions. But the more I learned, obviously, I recognized there were things that I wasn't paying attention to, or things that I wasn't fully thinking through, or that there were situations that I couldn't anticipate. And the real goal in all these things is to make sure that you have the impact you want, and taking the time to think about whether your intentions and your impact align, finding why there's a discrepancy and working towards minimizing that, I think is critical. But some of these larger skills, I think we have to build them into the education of people, because it becomes so important to their success and so important to these parts, which are sort of the more human side of the business of science.
Absolutely. Jason, anything to add? There was a lot there.
Oh yeah, I'll go with the latter part, which I don't think is going to surprise anyone who's listened to some of the podcasts I've done, is that what makes you a good grad student or a good postdoc or a good early PI is not what makes you successful long term, whether it's academia or industry necessarily. Like, some of it's there, right? Like your critical thinking skills and your ability to break down problems. But yeah, there's this big step function between individual contributor and leading a group of people, I think, that occurs everywhere. And I think science, and I'm going to say that outside of academia, because I think it's a problem in science generally, it happens in industry too. Science is not particularly good at building that natural on-ramp to go from an individual contributor to a leader of a team, whether that's postdoc to junior faculty, the senior faculty, or it's scientist one, scientist two to group leader, or what have you in industry. And so I think part of what we're getting at here is understanding the collaborations you set up and you negotiate with people is one of those group leader things that it's a lot more helpful if you get experience early. I know I was lucky in my postdoc. I got to set up collaborations and deal with data sharing and, okay, how are we going to publish this and who's doing what? So when it came to industry, and I knew what was a trade secret and what wasn't, I kind of had a template for how to go by it. But I also didn't have training on how to negotiate trade secret negotiations with academic labs and what we can share and what we can't. I had to go talk to legal for a little bit and figure it out.
Mhm.
And so I think the larger point that Alex makes about that learning curve and the soft skills is something that all of science would be benefited by creating a stronger development for, whether it's undergrad, grad school, postdoc, early career industry, early career academia. I don't think it matters. I think it's something where science is such an intellectual individual contributor pursuit early that you have a giant phase shift to the later phases, that there isn't a natural on-ramp that sometimes exists for other career paths to like build that up.
Yeah, I mean that's why we've done this podcast is exactly that. It's it's a mentorship podcast for a reason, because there are often no structured mentorship pathways to address some of these nuances. I want to get back to the data piece. Alex, I see you want to say something though.
Just
Let's
say
go ahead.
really quickly, even though, I mean, the thing to recognize is that different labs work on different problems or different sizes, have different interactions and connections to industry, to partners locally, across the state, across the country, across the globe, and it creates different opportunities. And so you'd like to see some greater normalization of those learnings and sort of a sharing. People are going to have different opportunities to practice it, but obviously people have different goals. And so you don't want to assume that everybody is going to want every piece of this, but you do want to have exposure. You were asking me at the beginning, why do I have a BA instead of a BS? It's because I went to a college to get a broad liberal arts education because I wanted broad exposure to things. And there are different pieces that I have different familiarity with and competency, and just because of how much I've used them. But you want to at least make sure that people have some of those tools, because once you have some of those tools, particularly some of these frameworks for thinking out some of these larger problems, it becomes much easier to work your way through stuff.
Mhm. Just for context, everyone, I was just double checking some of Alex's bio, and I thought it was a typo that he had a BA in physics, but he clarified that that was indeed what he obtained from Columbia. So that was the
Chemical physics, but yeah.
chemical physics, yes. Um, okay, I want to go back to open data. As much as I love talking about the gaps in mentorship in science, I could talk about that for hours. The piece that and there's no answers. Well, I won't say there's no answers, but is privacy, data privacy. And you spoke to this a little bit, Alex, when you talked about, respecting the sample providers, and that often these people are going through, certainly in what you're doing, they're going through the worst times in their lives when they're providing these samples, and there's a lot to consider there. But what are some of the maybe frameworks that are in place and some of the gaps around data privacy? And I will admit, I did my research on fish, and I was like, have at it. Here's the genome sequences of these fish. But that's a whole different thing when you're talking about people.
Oh, I mean, there's a lot to unpack here. There's a lot going on, and I think it really depends upon on who you want to include in this. And so, are we talking about open data with respect to sharing things within Boston, within the United States, around the globe, thinking about who is getting engaged as a collaborator and a donator of samples, and even more than that, like thinking about sort of the communication of consent, and what's in a consent, what do people really understand about consent, is the language interpretable in a way that they can really understand how the data will be used and reused. And there's a lot to unpack there and a lot to work through, and in many places it's easy to ignore it and to move forward, because you could say the hard part is the science. But I think that it's more important in a lot of ways to spend the time in the beginning thinking through these things and setting up structures that last. And I think, getting back to sort of the mentorship piece, I often tell people it's more important that you learn how to do science than that you accomplish specific things in science. And a PhD is really learning about how to approach the process and how to show up in the world to make sure that you are the kind of person and doing the kind of work that you want to do, more than guaranteeing any result, because those things are hard. And so open data becomes a big thing to wrap your head around. And I think that when we try and think about it through one specific lens, it becomes problematic, because there are differences that are readily apparent when you have conversations with people in different parts of the world. And I think that instead of trying to solve things with generalities, like really leaning in and thinking about how to partner with people and to create structures that work for them becomes the most important part of all of this. And you might say, well, it slows down the big part of open data. It doesn't let us do the mining on everything. And I would say, well, getting back to this idea of technical reproducibility versus biological interpretability, there are a lot of places where understanding how people did things, understanding the lens they apply to science, using that through the kinds of things you do towards interpretation and analysis can be incredibly empowering, right? So I think that building those partnerships builds the scientific community. It helps to advance people. It helps to identify opportunities when you pull data sets together. And so I think that open data is not easy. I think the process of working towards open data is almost more important in some ways than achieving open data, because I think, and a lot of people would argue with me on that, but I would say that the process of doing it and building the community and doing things right, getting people engaged, thinking about process, thinking about how to, really support and enable people and empower them, to me is the fulfillment of what we're really trying to do, as opposed to the thing that happens in the future.
Jason, anything to add?
So I think with privacy and consent, because I'm in charge actually of the privacy for our donor network at our company, that it gets very interesting. You made a point early on about, sometimes there's new things that we want to do with data that weren't even envisioned when the consent on the trial was obtained years ago. And we look at kind of the historical nature of consent. Like, frankly, it used to be terrible, right? Like, oh, here, sign here, don't explain it. You're good. We can do whatever we want forever. And so now we have a different reaction where you have to describe in detail every single thing you could possibly do now or in the future with the data and have an affirmative approval for that. And if you didn't list a type of test that didn't exist yet, you can never use the data for that in the future. And so then you get this really weird thing where you try to write broadly, but it could be so broad that it's meaningless, like all computational analysis used by but then that gets thrown out as being by IRBs as not being specific enough. but then you could then preclude the data from being open. So I wonder if part of the work may be coming to, and this gets back to standards, right? But like a more unified understanding of what true informed consent is and privacy protection, but putting privacy a bit aside of like anonymize the data, folks, start with that. Let's keep going here. And obviously there's some ways to back calculate out the lack of anonymity, but assuming it's anonymized data, to get that consent to allow the data to be used, knowing that 5 years from now, someone's going to want to use that genetic sequence for something that no one envisioned could exist now, and not completely losing access to all this data we had up to date because it wasn't listed the right way on the informed consent at the time. And I don't know if that's like a landmark event that needs to be aligned on.
Maybe, but it's one of those things where when you talk to people in other parts of the world, they would say the fact that you're even sharing genomic sequences, like that is something that you can't fully anonymize. And so, obviously there are places where we could think about like recontacting people. You could have consent to recontact, right? Becomes complicated because sometimes an individual unfortunately passes away. And then you think about, do you have the ability to recontact the family? And you also have to think about these larger questions of consent of the individual versus the community. In many of the conversations we've had with Indigenous communities, one individual may consent, but the way in which the information may be used may have an impact on the broader community. And it gets complicated as you start to think about, well, how do you do right, not just by that individual, but by the broader community that they represent? And that's where really thinking about consent becomes critical. But I would have argued that a lot of this comes back to the point that you made at the end about creating greater awareness and understanding of what consent is, what people are consenting for. And that comes back to being involved in greater partnership with these communities, having conversations, going back and forth, relaying what the science is teaching you, talking to them about what you're doing so that you have a common base for dialogue. And you could say it slows things down because it's impossible to do all of that, but it becomes important to sort of achieve the overall goal of what we're talking about here.
Mm-hmm. This relates back to another episode that we've done on this podcast about diversity and inclusion in science, and we talk about sample diversity and inclusion. And we could talk about so much more here. The hour has absolutely flown by, so I'm going to stop here and hope that we can have more conversations about this in the future. So thank you so much, Alex, and thank you, Jason, for joining us today. And thank you to all of our listeners, and we'll take care and see you again. That brings us to the end of our show. Don't forget to sign up for our email list at www.labcoatsandlifepodcast.com to get show notes, episode summaries, and links to useful information, or learn more about STEM mentorship via the resources found at www.stemcell.com/labcoatsandlife. You can also reach out to us on X via STEMCELL at STEMCELLTech or via email at info@labcoatsandlifepodcast.com with feedback or to suggest guests.